My, how things have changed in school since my time. I wonder if my theory of evolution by natural selection has made it onto the school curriculum. I certainly had problems with people accepting my ideas when I wrote it.
Nothing has been found to challenge your theory, Mr. Darwin, but there have been many attempts to discredit you.
To support your idea, we now have the discovery of the molecule of life, DNA.
In fact, we are doing an experiment today to show evolution in action through DNA.
You mean that you now do experiments to prove evolution in schools? Surely it's much too slow. You'd be in your graves before you got any results.
Of course, you haven't heard of microbiology and the amazing speeds at which bacteria can reproduce.
I did see some of the first photographs of bacteria. I guess they were going to be a real breakthrough in science.
Yes, we now use bacteria that naturally live in our gut to make drugs like insulin to treat diabetes.
That's amazing, but I want to know more about the ones that cause disease. My daughter Annie was killed by tuberculosis.
We now know that TB is caused by a bacterium, but we also have drugs called antibiotics that kill bacteria.
In fact, we are using antibiotics in our experiment today.
However, we have to be very careful that we do not infect ourselves with the bacteria.
That's why there was all of the hand washing, sterilising the benches with bleach, and flaming of the bottles.
I see. So, how are you showing evolution today?
We are looking at how bacteria can evolve by transferring antibiotic resistance genes between each other.
What's a gene?
It's the length of that DNA molecule we were telling you about. It's like an instruction for a cell to make something.
We are mating two different strains of E. coli bacteria that are resistant to different antibiotics.
We hope they will swap resistance genes. We will then have bacteria that have evolved resistance to both antibiotics.
Once we have mixed the two strains of bacteria together, we leave them in a warm incubator for 24 hours to conjugate. Let them get on with it.
We then plate the bacteria out on agar jelly. 2 plates contain the different antibiotics and the third plate contains both antibiotics.
Only bacteria that have successfully conjugated will be able to grow on the plates with both antibiotics because they need both resistance genes.
This is one of the plates with both of the antibiotics in the agar. You can see that only the bacteria that came from the mating broth are able to grow on it. Only they have the resistance genes to both antibiotics.
This animation shows how bacteria can exchange resistance genes. The resistance genes are on a small circle of DNA called a plasmid.
The donor bacterium forms a pillar to which the plasmid can travel down. The recipient bacterium is then resistant to both antibiotics as it already had a plasmid with a resistance gene on.
Thanks for bringing me up to speed, Brent, with my own theory. I'm very glad it still holds true in the modern world.
And thanks to all of you for watching this.